Hello, we are Team TTTF in Biochemistry 2. We are going to present about the citric acid cycle. Theme 1. Why is this chemical pathway called the citric acid cycle or TCA or Krebs cycle? It is called the citric acid cycle because the material produced in the first step of this cycle is citric acid. And we can also call this the TCA cycle because citric acid, which is the starting material, is a type of tricarboxyl acid. It means that there are 3 carboxyl groups. This pathway is also called the Krebs cycle because it was discovered by a British biochemist named Hans Adolf Krebs. Theme 2. Why is this chemical pathway so important in the metabolism of cells? The citric acid cycle is an important step in the respiratory process, which is generating an energy unit called ATP in the mitochondria of a cell. In this pathway, NAD+ and FAD are reduced to NADH and FADH2, and ATP produced in the electron transport system is used as energy for cells and is essential for maintaining life activities such as cell activity, growth, and division. This cycle also has important regulatory functions, such as interacting with metabolic pathways to produce important intermediates, participate in various biochemical reactions, and regulate energy balance in cells. As a result, citric acid cycles are known as essential chemical pathways for cell survival. Next is Theme 3. The citric acid cycle is composed of various intermediates and enzymes, making it complex. This complexity arises from its role in breaking down diverse biomolecules such as carbohydrates, fats, and proteins into energy. And it has a cyclic structure. This allows for the regeneration of intermediates, ensuring a continuous supply of substrates for energy production. So, a single starting molecule circulates multiple times, producing energy along the way. Lastly, it is efficiently designed. Through redox reactions, it generates energy and produces important intermediates for cellular functions and growth. Additionally, this pathway integrates various metabolic routes, facilitating the synthesis and removal of biomolecules. Theme 4. Summary of the cycle. There are 9 key intermediates in the cycle: acetyl-CoA, citrate, isocitrate, alpha-ketoglutarate, succinate-CoA, succinate, fumarate, malate, and oxaloacetate. Next, the changes in carbon number. In conversion of isocitrate to alpha-ketoglutarate, the carbon number is reduced from 6 to 5, producing CO2. And in the next step to produce succinate-CoA, the carbon number is reduced to 4, producing CO2. At the end, oxaloacetate and acetyl-CoA combine to produce citrate with 6 carbons, and the cycle repeats. There are 5 reversible reactions: citrate to isocitrate, succinate-CoA to succinate, to fumarate, to malate, and to oxaloacetate. And there are 3 irreversible reactions: acetyl-CoA to citrate, isocitrate to alpha-ketoglutarate, and to succinate-CoA. There are 5 locations of reactions where energy is produced or enzyme activity is controlled. The conversion to alpha-ketoglutarate, succinate-CoA, and oxaloacetate each produces NADH, and the conversion to fumarate produces FADH2. Lastly, the conversion to succinate produces GTP. Theme 5. Let's solve additional question 2. It is about the effect of FeCl3 concentration on yield during citric acid production using aspergillus niger. FeCl3 is ionized in aqueous solution and divided into Fe3 plus ions and Cl minus ions. Fe3 plus maintains the activity of these enzymes and ensures the smooth progress of the citric acid cycle. Therefore, if the concentration is out of the optimum value, the activity of these enzymes may interfere, negatively affecting the production of citric acid. When the Fe3 plus is higher than the optimum value, the aconitase activity is not maintained, making it difficult to convert citrate to isocitrate. When the Fe3 plus is lower than the optimum value, the isocitrate dehydrogenase activity is not maintained, making the conversion from isocitrate to alpha-ketoglutarate difficult. The chemical principle used in this process is Rule 5. Living cells utilize electrically charged functional groups to facilitate chemical reactions by lowering activation energy. Fe3 plus ions may be involved in maintaining the activity of the enzyme, and the enzyme promotes chemical reactions through a charged functional group when it is activated. Theme 6. The citric acid cycle. Step 2 is about the formation of isocitrate from citrate. The enzyme aconitase catalyzes this through the formation of cis-aconitate. Rule 10 is applied in two parts of the reaction. First, the enzyme aconitase removes water from citrate, forming cis-aconitate as an intermediate, and then adds water back to form isocitrate. This process is an example of the loss and gain of water by a carbon molecule. Both these steps occur without extra energy input and do not affect the overall oxidation state. This rearrangement of the hydroxyl group is needed to make the molecule more unstable, allowing it to obtain energy more efficiently. In this step, Rule 12 is also applied. This rule comes into play considering the actual concentrations of products and reactants in the cell. Even though the standard free energy is positive, which usually suggests the reaction is not spontaneous, the reaction proceeds in the direction of isocitrate formation. This is because isocitrate is immediately consumed in the next step of cycle, effectively reducing its concentration. Step 3 is about the formation of alpha-ketoglutarate, NADH, and CO2 from isocitrate. The enzyme isocitrate dehydrogenase catalyzes this through the formation of oxalosuccinate. This process occurs in two steps. First, isocitrate is oxidized by isocitrate dehydrogenase, transferring electrons to a carrier molecule NAD+, forming NADH. Rule 2 is applied in this step as an example of an oxidation reaction, specifically a dehydrogenation. Rule 7 is also applied to this step. The transfer of the acyl group from isocitrate to form oxalosuccinate is seen as an acyl group transfer, a common reaction between electrophiles and nucleophiles. This reaction forms oxalosuccinate as an intermediate. In accordance with Rule 5, the enzyme isocitrate dehydrogenase utilizes electrically charged functional groups to lower the activation energy of the reaction. Then, decarboxylation is facilitated by electron withdrawal by the adjacent carbonyl and coordinated Mn2+, releasing CO2 and resulting in alpha-ketoglutarate. The decarboxylation step involves the ability of the carbonyl group in oxalosuccinate to accept electrons. According to Rule 9, this ability is increased when it interacts with the positively charged manganese ion. Is the final oxidative decarboxylation, and this has similar mechanism to step 0. The reason is that the pyruvate dehydrogenase complex of step 0 and the alpha-ketoglutarate dehydrogenase complex of step 4 have similar structures and functions. The alpha-ketoglutarate dehydrogenase complex consists of 3 enzymes and 5 coenzymes. Alpha-ketoglutarate following Rule 5 due to the carbonyl group. Next, in accordance with Rule 6, this electrophilic carbon interacts with the nucleophile of E1-TPP. This interaction leads to the breaking of the carbon bond in alpha-ketoglutarate, releasing CO2. Subsequently, a new bond is formed between E1 and alpha-ketoglutarate. After forming the new bond, electrons are removed from E1's TPP, causing the disulfide in E2's lipoate to be reduced, transforming lipoate into succinyl lipoate. Then, CoA SH enters E2, following rule 7, leading to the transfer of the succinyl group to CoA. As a result, succinyl CoA contains a thioester bond, while lipoate accepts electrons. Reduced lipolysins undergoes dehydrogenation, an oxidation reaction according to rule 2, resulting in the loss of two high-energy electrons. These electrons are accepted by the FAD in E3, converting it to FADH2. Simultaneously, NAD+ enters E3, and the electrons from FADH2 are transferred to NAD+, generating the fully reduced electron carrier NADH. Step 5 is about the formation of GTP or ATP and succinate from succinyl-CoA. Firstly, succinyl-CoA undergoes hydrolysis, breaking the thioester bond. The energy released from this bond cleavage is then utilized to synthesize either GTP or ATP. The enzyme that catalyzes this reversible reaction is succinyl-CoA synthetase. Firstly, in accordance with Rule 7, the phosphate group is substituted for the CoA in succinyl-CoA. This results in the formation of a high-energy acyl phosphate. Subsequently, following the interaction between nucleophile and electrophile as Rule 6, the phosphate group is transferred to a histidine residue. Then, in succinyl phosphate, the phosphate group is released, resulting in the production of succinate. The histidine residue that received the phosphate forms a phosphohistidyl enzyme. Lastly, in accordance with Rule 7, the phosphate attached to the histidine residue is transferred to GTP, resulting in the production of GTP. Additionally, the reason the standard Gibbs free energy is only minus 2.9 kJ per mole is because step 5 is an energy-conserving reaction. Step 6 is about the formation of fumarate and FADH2. The enzyme succinate dehydrogenase catalyzes this reaction. In this reaction, succinate is oxidized by succinate dehydrogenase, transferring electrons to the electron carrier FAD, forming FADH2. Rule 2 is applied in this step as an example of an oxidation reaction, specifically a dehydrogenation. One thing to remember is that succinate dehydrogenase is an integral protein of the mitochondrial inner membrane in eukaryotes. Step 7 is about the hydration of fumarate to malate. The enzyme fumarase catalyzes this reaction. In this step, the reversible hydration of fumarate to L-malate is catalyzed by fumarase. The transition state in this reaction is a carbon ion. This is a process where a water molecule is added to the fumarate molecule. Specifically, the hydroxyl group from water is attached to one carbon atom, and the hydrogen atom from water is attached to the other carbon atom. According to Rule 10, this reaction does not require extra energy input because it's a simple addition of water. Moreover, fumarase is highly stereospecific, meaning this enzyme only catalyzes the L-malate, not D-type malate. Step 8 is about the formation of oxaloacetate and NADH. The enzyme malate dehydrogenase catalyzes this reaction. In this reaction, L-malate is oxidized by malate dehydrogenase, transferring electrons to the electron carrier NAD+, forming NADH. Rule 2 is applied in this step as an example of an oxidation reaction, specifically a dehydrogenation. In the citric acid cycle, this reaction has a high standard free energy change, which means it's not spontaneous under standard conditions and would naturally tend to proceed in the opposite direction. However, according to Rule 12, the actual direction of a reaction depends not only on the standard free energy change, but also on the actual concentrations of the reactants and products. In this case, the subsequent reaction catalyzed by citrate synthase, which is involved in the first reaction of the citric acid cycle, keeps the concentration of oxaloacetate very low. Since the first reaction has a very large negative value of standard free energy, the sum of the less than first reactions is negative, which causes the reaction to proceed. So, this is the role of team members in TTTF, and these are references. Thank you for watching this video.